Nanobiomaterials. That means the biomaterial in nano state. You know what is biomaterial? We have studied biomaterial elaborately. We have studied a chapter of biomaterial, which is used in our body. The material used actually in our body, they are biomaterials, and they are biocompatible also. Do you know what is biocompatible? We have discussed it earlier. What is biocompatibility? Biocompatibility means the materials which are compatible to our body. That is biocompatibility. So the biomaterial in nano state is known as nanobiomaterials. That means nanomaterial used in our body. The nanomaterials used in our body. We have seen when we studied nanomagnetism. When we have studied nanomagnetism, we have seen some nanomagnetic material which have biological activity. That means biological activity means we have used them in our body for MRI, magnetic resonance, for MRI. That means the magnetic the nano ferromagnetic materials, they shows the super paramagnetism. They can be used to image the some parts of our body. So that ferromagnetic materials, that nanoferromagnetic materials is a biomaterial, but not all the nanomagnetic materials is biologically compatible or biocompatible. Only the iron compounds are the biocompatible, because we have iron in our body in nanostate. So iron compounds are your biocompatible, and they can be used as materials. There are many other elements or compounds or something. They in nanostate, they are biocompatible. We shall study them. We shall study about them now. So 7:12 হয়ে গেছে, এখনো লোকজন আসছে না। আমি এটা বন্ধ করে দিই, তাহলেই লোকজন আসা শুরু করবে। So nanobiomaterial or nanobiology. Nanobiology. What is nanobiology? The synergy of the surface science and molecular biology has given birth to a new subject called nanobiology. The synergy of surface science, synergy and molecular biology. Surface science means nanoscience. Surface science means nanoscience because nanomaterials has the surface atoms only. In maximum nanomaterials, you will find the surface atoms only. For that, it is sometimes it is called surface science. And molecular biology. You see, in our body, if you think about the condition or about the working of our body, you will see that everything is nano. Say for example, your cell. Your cell is not nano, but inside your cell, say for example, DNA is working, mitochondria is working, they all are nanomaterials. The the all are the small things. All are the small things that are working in our body. So scientists wants to see how this nanomaterial is working in our body. You see, a DNA. DNA is very small. Now, what is nanomaterial? Nanomaterials means is not that one molecule or one atom. It is a nanomaterial makes of a number of atoms, which has a length of say 10 to 100 nanometer. If you collect a number of atoms which have a length in breadth or in width, which have the length of, say, for example, 100 nanometer, from 10 nanometer to 100 nanometer, we will call that nanoparticle. Not one atom is nanoparticle, or one molecule is not a nanoparticle. It is a collection of atoms. Say, for example, 50 atoms. You have 50 atoms, you have taken 50 atoms, and this 50 atom has a length of say 50 nanometers. So all these 50 atom is known as a nanoparticle. So if you see a DNA, say a DNA has a length of say 75 nanometer. 75 nanometer. So a DNA has a length of 75 nanometer. So a DNA is a nanomaterial. If a DNA has a length of 75 nanometer, so a DNA becomes a nanoparticle. If a mitochondria has a length of say 50 nanometer, then it becomes a nanomaterial. So nanomaterials in our they are working in our body. Otherwise, it will not be possible. If they are not small, our body cannot function. It is a very complicated. It is a very complicated thing. It is a very complicated thing. Can you imagine? Say, for example, a kidney. A kidney is like like of this length. This length, but it is working huge. If you see the dialysis process, if you see the dialysis, the machine is huge. That means dialysis is the substitute of kidney function. If your kidney is not working properly, you do dialysis, and the dialysis machine is huge. The work of that huge machine is done by this size of your kidney in our body, and properly. So everything, say for example, our heart. What will be the size of our heart? At best this size. But how complex is its working? they all are working in nano level. They all are working in nano level. So, the scientists, the scientists are very eager to see how these things are working and how they can reproduce it in the laboratory. So the synergy of surface science, that means the combination of surface science, that means the combination of nanoscience with molecular biology. Why it is called molecular biology? Because this works at molecular level. This works at molecular level. So that's why it is called as molecular biology. So molecular biology, that means it works at molecular level. That means it is a small thing, and the nanoscience is a small thing. The molecular biology is a small thing, and the nanoscience is also a small thing. So if you combine these two, both the small things, if you combine both the small things, say for example, a DNA is very small thing. If you combine a gold nanoparticle with that DNA, that will be a nanoparticle. So if we can use a gold nanoparticle, combination of gold nanoparticle with DNA for some use, it will be called as nanobiology, and the material is known as nanobiomaterial. So the synergy means combination. The synergy of surface science and molecular biology has given birth to a new subject called nanobiology, which symbolizes a path-breaking evolution in the progress of biology. That means it symbolizes, it does a path-breaking evolution. Say, for example, genetics and genetic engineering. It is, it is the progress of biology. Genetics is the progress of biology. Your pharmacy is the progress of biology. So, 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 uh, uh, so this nanobiology actually actually creates a new field to the biology as well as nanoscience. So this new subject is capable of unveiling many fundamental secrets of life forms. The science of nanobiology has progressed significantly and can be branched into several disciplines of life science, such as cellular biology, genomics, proteomics, oncology, immunology, diagnosis, targeted drug delivery, etc. You know, this nanobiology, this nanobiology works on different fields of biology. You see, what is it is cellular biology. That means cellular biology works on cell of our body. Genomics means it works on genes of our body. Proteomics means protein related science. You know, our body is made up of protein. So this is protein related science, and you see the protein is also a nanomaterial. Everything which have a length of 10 to 100 nanometer, it is a nanomaterial. So oncology means in cancer detection. Immunology, you'll know what is immunology. It deals with our immune system. Diagnosis That means diagnosis of our malfunction of our body, and a targeted drug delivery. That is very important. Targeted drug delivery. The drug is delivered on the place, at the place where the problem is found. The drug is delivered. If you have a pain in your knee, if the drug is delivered in the knee itself, not on other parts of the body, then it is targeted drug delivery. So nanobiology works works all these fields, in all these fields. So the idea of creating the hybrid system of inorganic nanoparticles with biological moieties. So in nanobiology, there is a hybridization of inorganic nanoparticles. Inorganic nanoparticles means, say for example, iron, say for example, gold, say for example, silver atoms. They are inorganic atoms, or say for example, iron oxide, say for example, cadmium selenide. They also are inorganic nanoparticles. So mainly nanoparticles are inorganic. Why they are inorganic? Because they are small. The organic molecules, the organic molecules are big, and they have different types of atoms with them. An organic molecule is a big molecule. They have different types of atoms with them, and these different types of atoms attracts another atoms by different ways. As for example, they have polarity, they have van der Waals attraction, they can have hydrogen bond, different types of bond they can they can produce because they have different types of elements. So they will agglomerate between themselves and make a big sized atom which is not nano. So mainly inorganic materials are the nanoparticles, but there are always some organic materials, and even you will get the polymer nanoparticles also. But mainly there are inorganic nanoparticles. So the the idea of creating hybrid systems with inorganic nanoparticles with biological moieties has helped to solve several technical difficulties in medical and biosciences. If you hybrid, if you mix inorganic nanoparticle with a biological moiety, say for example with protein or with DNA, then it will help to solve several technical difficulties in medicals and biosciences that medical science cannot cannot solve a problem alone with these biological molecules. But when you when you hybridize an inorganic particle with that biological molecule, they can solve the problem. Using nanomaterials instead of conventional materials in life science applications have increased efficiency while decreasing cost. If you use nanomaterial instead of conventional material, then then efficiency of the material will be increased, as well as it will decrease the cost because the nanomaterial has a small amount. The material and nanomaterial requires a small amount, so it will decrease the cost. Thus, the hybridization of nanobiotechnology has laid the ground for many novel methodologies. The ideas and innovations of nanobiology can be extended to the frontiers of biotechnology. This this hybridization and the hybridized product and use of this hybridized product is 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 produced a new type of science which is known as nanobiotechnology. The hybridization of what inorganic nanomaterials with biological system and use of this inorganic nanomaterial with biological system to our body leads to a new uh, subject which is known as nanobiotechnology. So there is a difference between nanotechnology and nanobiotechnology. Nanobiotechnology works in our body, and nanotechnology can work all over all in other areas. So you see, for example, this is a inorganic inorganic nanomaterial. For example, let us suppose it is a gold nanomaterial. It is a gold nanoparticle. By telling about the gold nanoparticle, it means that there are it is not one atom of gold. Again, I'm telling you, this is not one atom of gold. This is a collection of gold molecules which have a nano dimension. That may say, for example, this is a quantum dot. If it is a quantum dot, if it is a gold quantum dot, its height, length, and width all are in nano dimension. So for example, this is a gold quantum dot. And with this gold quantum dot, with this gold nanoparticle, you can hybrid this gold quantum dot with drugs. You can hybrid with gold quantum dot with dendrimers. You can hybrid with gold quantum dot with antibodies. You can hybrid the gold quantum dot with DNA, or you can connect the gold quantum dot with carbohydrate. You can connect with fluorescent dyes. You can connect with peptides. You can connect with some markers. They they can be connected with several biological molecules. These all are biological molecules. These all are biological molecules. So you can connect the inorganic nanoparticle as you wish. If you want to connect with DNA, you can connect with gold nanoparticle with DNA, and you will have an application of gold nanoparticle with DNA. You can connect with antibodies with gold nanoparticle, and you have that application of gold nanoparticles with the antibodies. So the main thing is in the nanobiology, what you have to do, you have to take a nanomaterial, a nanoparticle, and connect it with a biological molecule. Then you will get a nano biomaterial. You'll get a nanobiomaterial. And you can use that nanobiomaterial in different fields. We'll see some of the fields later, but you can use it in different fields. Now, what are the major aspects of nanobiology? First one is interaction between biomolecules and nanoparticle surfaces. You see, this is a nanoparticle, and the interaction of biomolecules, say for example, interaction with DNA or interaction with carbohydrate, where they will interact with the surface of the nanoparticle, not in other places, not inside the nanoparticle. So there will be an interaction of carbohydrate molecule with the surface of this nanoparticle. So if they interact, if the surface of this nanoparticle interacts with this carbohydrate, then what will happen? This is the this is one of the aspect of your nanobiology. One of the major things which we have to study in the nanobiology. Then biological imaging using nanoparticles. You can image a biological object. You can see a biological object by using nanoparticle. Say, for example, you want to see the size of a tumor in your body. You can see it by means of nanoparticle. So this is one of the aspects of nanobiology. Analytical application of nanobiology, that means you can analyze a biological object. Say, for example, nowadays we are hearing about the different variants of coronaviruses. You'll see there are some variants of South Africa, there are some variants of Brazil, there are some variants of UK of coronavirus. Now, what is the variants? What is the variant? It is that a virus has a DNA strand, or it has a RNA strand, and there is a sequence of DNA or RNA. Say, for example, in a DNA, there are four bases: adenine, guanine. What are they? Adenine, guanine, uracil, and cytosine. Not that, is it? Adenine, guanine, uracil, or cytosine. Whatever it means, A, B, C, D, say, for example. There is a sequence of base A, B, C, D. And the next sequence is C, D, A, B. And the next sequence is D, A, B, C. But if somewhere the sequence is break, say, for example, it should be D, A, B, C. Instead, it becomes D, B, C, A. The sequence is changed in one position. That is called a variant. That means the coronavirus, this is the coronavirus strand. If the sequence, DNA sequence in a place is changed, that is known as a variant of coronavirus. In UK, the sequence is changed. In South Africa, the sequence is changed. So the South Africa, that is called South Africa variant, and the UK variant and the Brazil variant, that are variant. Now, how to check this variant? How to check this sequence? You can analyze DNA by nanobiological technique, by nanobiotechnology. So this is one of the aspects of nanobiology. We'll see it later in a small extent, how you can check the sequence of a DNA. We'll see it later. Medical diagnosis, that means diagnosis of your some problems, diagnosis of your body problems, and I have told you what is targeted drug delivery and biosynthesis of nanomaterial. How can you synthesize biologically nanomaterial? These are mainly studied on the subject nanobiology. These are the major aspect of nanobiology. That means these things are mainly studied under the nanobiology. Now, first thing, interaction between biomolecules and nanoparticle surfaces. To go on this, before going on this topic, first I have to ask you: is there any question of you? কোনো question আছে? কোনোটা বোঝা নাই এমন কিছু আছে? নন্দিতা কোথায় গেলে? আছো কি তুমি? নাই তো দেখি। কোথায়? খাদিজাতুল কোবরাও তো নাই দেখি।
স্যার ওর কারেন্ট চলে গেছে, আর
ঢুকতে পারতেছে না? দাঁড়াও আমি খুলে দিচ্ছি। তোমরা এমন করো যে মানে আমাকে এগুলো করতে বাধ্য করো তোমরা। আমি 7:12 পর্যন্ত তোমরা আসছো না, তোমরা যে যখন খুশি তখন আসবে। এই যে দেখো নন্দিতা কোথায় চলে গেছে। আচ্ছা, তোমাদের কোনো কোশ্চেন তো নাই না? Very good. So, interaction between biomolecules and nanoparticle surfaces. Now, what we have to do first, to prepare a nanobiomaterial, you have to take an inorganic nanomaterial, and you have to hybrid it. You have to mix it. You have to attach it with a biological molecule. Say, for example, you have to attach it to the protein. So, you have to first take a suitable nanomaterial, suitable inorganic nanomaterial, which is biocompatible. You will get many nanomaterials. You'll get many nanomaterials, but they are not biocompatible. Biocompatible means what? It doesn't do it. It do not make any harm inside our body. Say, for example, iron or iron oxide is a very good biomaterial. It is biocompatible. Gold is very much biocompatible, and silver is also very much biocompatible. We generally take these three atoms as our nanobiomaterials to study our nanobiomaterial. So if we take, say for example, if we take a gold nanomaterial, then you will see that the surface of the gold nanomaterial is of gold 1, that means gold plus, and the inside atoms of gold nanomaterials is of A0, that means neutral gold atom. So a gold nanomaterial is made of gold with two oxidation states. In surface, it is +1, and inside it, the core gold material are A0, AU0. That means the oxidation state is 0, because because in the surface generally gold 1 is absorbed, and inside it it becomes gold 0. Now a gold nanomaterial, that means a gold nanomaterial is positively charged. Now if you treat that gold nanomaterial with any citrate, ammonium citrate, you can use sodium citrate, you can use potassium citrate, you will get citrate ion. So citrate ion is negatively charged, but it is 2 minus or 3 minus. So when the gold nanomaterial is interacted with any citrate ion, then the gold citrate compound or the gold citrate complex is negatively charged as a whole, because gold has only one positive charge, but citrate ion has a -2 charge or -3 charge. So gold and citrate, when you mix gold and citrate, when gold and citrate interacts with each other, then the gold citrate complex is negatively charged. Now, the proteins, say for example, a biological molecule, the protein has a unique property which is known as isoelectric point. Do you know what it is? Isoelectric point. Have you studied protein in 4th year? তোমরা পড়েছো protein 4th year?
Yes, sir.
ওখানে isoelectric point পড়ো নাই? So proteins have a isoelectric point. What is an isoelectric point? Below this point, that means below this point means pH. Below this pH, a protein has a positive value, and above this point, the protein has a negative charge. That means isoelectric point is a point when the pH of a protein sample solution is below the isoelectric point, the protein have a net positive charge. That means if the pH of a protein is below the isoelectric point, then the protein will have a positively charged, and if it is above the isoelectric point, then it will have a negative charge. So at isoelectric point, protein will have a charge 0. Below the isoelectric point, protein will have a charge positive, and above the isoelectric point, the protein will have charge negative. So, what is isoelectric point again? I'm telling you, isoelectric point actually is a pH. Say, for example, in a definite kind of protein, say, for example, alanine is a protein, and the isoelectric point of alanine is at pH 4. That means at pH 4, the protein is neutral. Above pH 4, the protein is negatively charged, and below pH 4, that is pH 2, pH 3, pH 1, the protein becomes positively charged. This is the isoelectric point. So, what happens? A gold citrate nanoparticle, we have prepared a gold citrate nanoparticle which have negatively charged. And if I place that gold citrate nanoparticle in a protein solution which have pH below isoelectric point, then the protein solution, the proteins will be positively charged, and the protein will attract the gold citrate complex. Okay, have you understand? The protein is positively charged below its isoelectric point. We have make a solution of protein which have pH below its isoelectric point. Say, for example, alanine has isoelectric point 4. For example, it is not true. For example, alanine has isoelectric point 4. And we have made the protein solution at pH 2. That means we have kept the protein in a solution which have pH 2. Now this protein becomes positively charged. And if I now treat this protein with this above gold citrate complex, which is negatively charged, then the protein will attract the gold citrate complex to make a gold protein nanoparticle. This is the interaction between biomolecule and nanoparticle surfaces. So it is very much pH dependent. That means it is very much not pH, it is very much isoelectric point dependent in case of protein. It is very much isoelectric point dependent. If you make this protein solution above isoelectric point, then the gold nanoparticle will not attract the protein molecule. Will not attract the protein molecule. So this thing is very much related to the isoelectric point. So this is a figure. This is a figure. You see, this is a gold nanoparticle, and this is a gold nanorod. You see, it is a quantum dot, or it is a quantum rod or nanorod. When it is treated with protein, then you will find this type of gold nanoparticle. Gold nanoparticle, gold protein bio nanomaterial. Gold protein bio nanomaterial. Now, it can be used in the diagnostic labs for early detection of diseases, and it can be used in therapeutics for the targeted delivery of nanoparticles. It can be used to targeted delivery in the nanoparticle. How they can be used, we will see it later. But this is the process of making a gold or any nanoparticle with a biological molecule to form a nanobiomaterial. Now, there are some electronic effects of biomolecule nanoparticle interaction. They attracted each other. Who? The gold nanoparticle and the protein attracted each other. Then what will happen? When a biomolecule has interacted with the surface of a noble metal nanoparticle, its surface charge density is deformed by electrostatic attraction. The surface charge density of gold nanoparticle will be deformed by this electrostatic attraction of what biological molecule. How it is deformed? What will be this the extent of deformation? This extent of deformation or the surface charge density, deformation of surface charge density depends on the following factors. What are they? The size of the noble nanoparticle, either it is 100 nanometer noble nanoparticle or 10 nanometer noble nanoparticle, or it is 15 nanometer nanoparticle. The molecular diameter of the biomolecule, it may be protein, or it may be carbohydrate, it may be your DNA. Then the diameter is changed. So depending on the diameter, the surface energy density also will change. pH of the medium, it is very much important. I told you the pH of the material, the pi value depends on pH. So the pH of the medium is important, and the ionic strength of the medium. In the medium, how much is the ionic strength? On the basis of these four factors, the surface charge density of a gold nanoparticle, the deformation of surface charge density of gold nanoparticle will um, mm, will be will actually it will depend on these factors. Now, when the nanoparticle or and the biomolecule attracts each other, the surface charge density actually perturbed. This perturbation of surface charge density, perturbation means your deformation. Perturbation means changed. Perturbation means deformation. Changed it is. This perturbation depends on the surface electron density of nanoparticle. That means whether it is spherically symmetrical or it is like a nanorod, whether it is like nanorod or it is spherically symmetrical. If it is spherically symmetrical, the deformation is generally isotropic. Isotropic means the deformation occurs occurs your similarly in everywhere. That is called isotropic. If the gold nanoparticle or if any nanoparticle is spherically shaped, then the deformation is regular, or the deformation is equal in all the sites. But if it is like a nanorod, then the deformation is anisotropic. You know what is anisotropic? It is direction dependent. So, in case of novel metal nanorods, their charge density is anisotropic. Thus, the interaction with the biomolecule can lead to anisotropic perturbation of electron density at the nanorods. So, this deformation of surface density, it can be isotropic or it can be anisotropic. It will depend on the size of the gold nanoparticle. Now, one more thing, the the biological molecule, the biomolecule that is attached to the nanoparticle, the size of the biomolecule that is attached to the nanoparticle, depending on the size of the biomolecule, the agglomeration is also dependent. Agglomeration means one nanoparticle is attached to another nanoparticle, which will be attached to another nanoparticle. That is agglomeration. If agglomeration is there, high amount of agglomeration is there, the particle will not be nano, it will be a bulk material. So, the natural agglomeration should be small to make a nanoparticle. You see, it is the actual image, TM image. TM image means transmission electron microscopic image. It is the TM image of a cysteine capped gold nanomaterial. That means cysteine. Cysteine means cysteine is a one kind of protein. Cysteine with gold nanomaterial. And it is vancomycin. Vancomycin is one kind of antibiotic. So gold is attached with vancomycin here, and gold is attached to cysteine here. The size of the cysteine is small, so there is a high amount of agglomeration here. But the size of the cysteine, vancomycin is large. So you see there is a small amount of agglomeration. They are separated from each other and they are truly nanomaterial. To make a nanomaterial, you must have separate them at nano size. If you agglomerate, it will be a bulk material. So the size of the biological molecule is also important. If the size of the biological molecule is small, the tendency of agglomeration increases. But if the size of the biological molecule is large, the tendency of agglomeration decreases, that and it will be a pure nanomaterial. So this is the main thing of the main thing of your interaction. What is that? Interaction between biomolecules and nanoparticle surfaces. Now, do you have any questions? কোনো কোয়েশ্চেন আছে তোমাদের? বুঝেছ পুরাটা? কোনো কোয়েশ্চেন আছে? আজাদ?
স্যার আইসোইলেকট্রিক পয়েন্টের ব্যাপারটা যদি আরেকটু স্যার
Isoelectric point. Isoelectric point is a pH. Say for example, আমি তোমাদেরকে আবার বলছি, say for example, alanine. Alanine is a protein. It has an isoelectric point at pH 4. That means at pH 4, alanine has a charge 0. It is neutral. Below pH 4, it has a charge, positive charge. And above pH 4, it is negatively charged. So the point at which the point means the pH at which the protein molecule is neutral, that pH is known as isoelectric point, and it is written by pi. By not writing H, it is written by pi. That is called isoelectric point. It is a very important thing of protein. তোমরা কিভাবে মিস করেছো আমি জানি না, but it is a very important thing of protein. আর কারো কোনো কোশ্চেন?
আইসোইলেকট্রিক পয়েন্টটা তো স্যার বুঝছি স্যার, ওখানে স্যার ওই যে ন্যানো ম্যাটেরিয়ালটার চার্জটা স্যার ওখানে
ওইটা বুঝতে পারছো না। আমি যে গোল্ড ন্যানো ম্যাটেরিয়ালটা বানালাম, হ্যাঁ, a citrate gold complex, it is negatively charged. Now আমি যদি সেটার সাথে প্রোটিনকে আমি যুক্ত করতে চাই, অ্যাটাচ করতে চাই, what I have to do? আমাকে কি করতে হবে সেটা বলো।
পজিটিভলি চার্জ করতে হবে স্যার,
পজিটিভলি চার্জ করতে হবে। তো পজিটিভলি চার্জ কখন হবে? when
পিএইচ যদি কমায়।
হ্যাঁ হ্যাঁ, তাহলে আমাকে পিএইচটা কোথায় কি করতে হবে? বিলো আইসোইলেকট্রিক পয়েন্ট আমাকে রাখতে হবে। Then if I add that gold nanoparticle to the protein, then the protein will attract the gold nanoparticle and will find a nanobiomaterial of gold and protein. যদি আমি পিএইচটা বাড়তি হয় অথবা পিএইচটা আইসোইলেকট্রিক পয়েন্টের সমান হয়, then I cannot find a gold biomaterial. বুঝতে পেরেছো জিনিসটা?
Yes sir, thank you.
আর কারো কোনো কোশ্চেন? আর সবাই সব বুঝেছো? The interaction of nanomaterial with biological molecules is heavily dependent on those four things. কি কি বলেছিলাম? The size of nanomaterial, the radius of your biomolecule, the pH, and the ionic concentration of the solution. On that fourth thing, the addition of nanoparticle and biomolecule depends. Now, যখন তুমি add করবে, when you add that biomolecule with that gold nanoparticle, the surface of the environment of the surface of nanomaterial will be perturbed. That means will be disturbed by the biological molecule. এই যে ডিস্টারবেশন, এই যে ডিস্টার্ব, এইটা কিন্তু this will affect on gold nanomaterial. কি এফেক্ট করে? এটাকে বলা হয় plasmon resonance. What is plasmon resonance? Plasmon resonance হচ্ছে ওই গোল্ড ন্যানোম্যাটেরিয়ালের তোমার যে সারফেসে যে ইলেকট্রন গুলো থাকে, they generally oscillate with the positive charges. This oscillation will be disturbed. The electrons in the surface of the gold nanomaterial generally oscillates with the positive charges in the present in the gold. This oscillation will be disturbed when a gold, a protein or a biological molecule you're attached with the gold nanoparticle. From this disturbation, we can characterize whether the protein is attached with gold or not. যে অসিলেশন হচ্ছে, এরকম একটা অসিলেশন হচ্ছে, যখনই প্রোটিনটা গোল্ডের সাথে অ্যাটাচ করলো অসিলেশনটা এমন এমন শুরু হলো। তাহলে এরকম যতক্ষণ অসিলেশন হচ্ছে আমি বলছি কি প্রোটিনটা এর গায়ে লাগে নাই। যখনই অসিলেশনটা এমন এমন শুরু করলো তখনই বললাম যে গোল্ডের সাথে প্রোটিনটা লেগেছে। কারণ গোল্ডের সাথে প্রোটিনটা লেগেছে কিনা এটা তো আমি চোখে দেখবো না, because they are nano, they are above my eyes capacity. How can I characterize? আমি কিভাবে বলবো যে এটার সাথে গোল্ডের সাথে এটা লেগে গেছে? I shall not get anything. তখন আমাকে কি করতে হবে? ওই অসিলেশনটা মাপতে হবে। ওই অসিলেশনটা যদি জেনারেলি গোল্ড ন্যানোপার্টিকেলের একটা অসিলেশন থাকে, এই অসিলেশনটা আমরা আমাদের একটা রেকর্ড করা থাকে। যখনই প্রোটিন ওটার সাথে অ্যাড হবে তখন অসিলেশনটা চেঞ্জ হয়ে যাবে। যখনই ওই অসিলেশনটা চেঞ্জ হবে তখনই আমরা বুঝতে পারবো যে প্রোটিন গোল্ডের সাথে অ্যাড হয়ে গেছে, আদারওয়াইজ অসিলেশন চেঞ্জ হতো না। That is called surface plasmon resonance, and the spectroscopy regarding this oscillation, change of this oscillation is known as surface plasmon resonance, your spectroscopy. This is the interaction of your surface nanoparticle surface with biological molecules, surface of nanoparticle with biological molecules. আজকে পর্যন্ত থাক। সামনের দিন আমরা আরেকটু ডিপে যাব, আরেকটু গভীরে যাব, ন্যানো বায়োটেকনোলজি। আমরা আরেকটু গভীরে এগুলোতে যাব।